Now finally we are here to talk about genetic crosses, and we're going to start by doing the true breeding cross, which is the first cross of Mendelian genetics, which is the first step that he did to make sure that the flowers he started off with were pure bred true breeding flowers that can only make more copies of themselves, like we talked in the previous video. Now what we're going to be using throughout this video, pod color as or character. And for pod color, Mendel used big G, and which to represent green, which was the dominant, which was the one that always overpowered, little g, which was representing yellow, right? And the genotypes therefore are big G with big G, which is the homozygous dominant, or big G with little g, which is the heterozygous or the hybrid, and then the little g, which is the homozygous recessive. Now in this, there's two ways to do a purebred cross. You can do little g versus little g, or you can do big G versus big G. Either way, it's going to look just like this. You're going to get big G versus big G for the genotype. So I'm crossing two flowers which are pure and have exactly the same genotype. That means I'm crossing a green flower with the green flower. Now, I'm going to do meiosis here and separate the alleles which are in these stains to create my gametes. How do you do that? You put one gamete of one pair in one line and then one in the other. So you're going to be creating one G, and then the other parent can only donate a G or a G. So those are the possible gametes. And now you distribute those gametes to each side of the Punnett square. So one parent you put up here, so you put one gamete, then the other, and the other parent you put to this on the side. Then you just combine those, you're doing fertilization, and you make the actual thing. So as you can see, the whole Punnett square is going to be the same thing. When you cross a flower that only has big G's, all it can do is big G children. And that's what we talked about in the previous video. So you're going to get, the only genotype you're going to get is big G, which means the ratio is 100% big G, homozygous dominant. The phenotype is going to be, there's only one code, so there's only one phenotype. It's going to be all green, and the phenotype ratio would be 100% green. Now what that means is that when you have all dominant parents, all dominant children will be the result, which means if you only have one type of allele, only one type of offspring is possible unless a mutation happens. And so this is the conclusion of the true breeding cross, and it's a good example to a simple thing to do this. Now if you did little g's, everything would be little g's in yellow. It would be the same thing. So I'm not going to spend any time doing that. Now remember that a self-cross of pure breeding specimen will always be like this, proving that they are actually true breeders. That's what true breeder means. If you do self-pollination, we will always do this. And this was very important for Mendel's experiment because he wanted to make sure that his first generation, his parental generation, was like this. So once he made sure that those flowers were truly like that, what he did is he got a true breeding green pod and he combined with a true breeding yellow pod, for example. So let's look at the plots here. And he actually found out that when he did that, all of the children came out green. And so let's talk about what happened there. And that's how he figured out what was going on. So, again, the pod color is going to be the same thing as we did before. Nothing changes here. The character, the alleles, the genotypes are all the same. But except this time, he's going to call it a pure breeding green pod. What is that? Think about it. Write it down. Pause it. I encourage you to pause throughout this video as before I actually click, so you have a chance to actually try yourself. So true breeding green flower is going to be big G. What about a true breeding yellow flower? It's going to be big little g. Now what that means is that the first flower is going to be green and the second flower is going to be yellow, just like it shows up there. Now you're going to do meiosis here and separate the alleles, big G, or the other one can donate a big G, a little g, and a little g. For some reason the color is not right here. Let me fix that. Alright, so you have the same setup, and now you have the meiosis done, and everything's back to normal. Now, you do the same thing you did last time. You distribute the alleles to each box. So on the top, you're going to put one parent. So you put one parent on the top, and each allele goes in one box, and you do the same thing on the side, and there you go. Now, you just combine the fertilization. Now, if this allele, this gamete meets with that gamete, what do you get? Big G, little g. What if this gamete meets with that gamete? You're going to make the same thing. What if this gamete, and so forth. As you can see, That is why you got all green flowers, because no matter what happens, every time in the Punnett square, you get a big G, little g, showing that when you duke out a pure green dominant versus a pure recessive, the result is a hybrid. That's the term for hybrid, what comes out of a parental cross. This is called a P1, or the parental cross, and the result of it is 100% big G, little g, which means since the dominant look always is present when the dominant gene is there, all of these plants will have green pods, and it will be 100% like that. And so now what that means is that in the P generation, which are the progenitors, are going to be two different kinds of true breeders, and they're going to make the F1, or the first generation, and they're going to be all progeny, all right, of all hybrids. And that's how this works. Now, this is the best cross to show the law of dominance, because it's the only cross that puts two genotypes which are pure against each other, and the offspring will show whoever is the dominant. When you put two pure breeds versus each other, the offspring will look like the dominant parent, because all hybrids are dominant. So this is the best cross to show the law of dominance. All right. However, remember, there's something we're going to learn about later. If it's called incomplete dominance or codominance, it will change this. In incomplete dominance, there is no such thing as a dominant look on the hybrid. The hybrid actually blends. The way we said it didn't happen, it actually happens. Some traits do that. And we're going to talk about that later. And there's also codominance, which is even cooler, when both traits show up at the same time in the hybrid. So hybrids have different relationships, and we're going to talk about that in the end of the genetics lecture. Now remember that the conclusion for this cross is that when crossing two different true breeders, all of the offspring will be hybrid, and that the hybrids will look like the dominant parent, which means you can use this to figure out who's the dominant. All right? That means that if you cross two of the same true breeders, you always get the same true breeders. That's the true breeder cross. But if you cross two different kinds of true breeders, you're going to get all hybrids, and the hybrids will all look like one of the parents, which is the dominant parent. All right? Now, on the next video, we're going to be doing the F1 and the F2 crosses. All right? Which is getting these children and crossing them themselves. We'll see. Now, what you see here is an actual example of something like that outside of flowers, because I want to keep you realizing that this doesn't only work for flowers, it works for anything. In this example, you get a fruit fly that has red eyes, and a fruit fly that has white eyes, and you combine them, and all the flies have red eyes. What does that tell you? It tells you that if that red was a red flower eye, that was a true breeding red flower, that means when it crossed with itself, if that was even possible, it always made itself. What that's telling you is that all that red is dominant over white. And so that's why it's the best cross to show dominance. And that's another example of this same cross I just talked about. So in the next cross, I'm going to be doing the F2 and the F1 cross, which is going from here on. So I'll see you there.